of the sterile tissue. Which one is more? In the which sporophyte is more? Based on that structure, here two theories are there in the explanation of the evolution of sporophyte in bryophyte. Okay, clear? Based on
Yes,
the
sir.
sterilization means sterile tissue complexity or structure of the sporophyte. Sterile tissue formation or either structure of the sporophyte or complexity of the sporophyte. So based on these development here, the explain the sporophyte. Here two theories are there. So in the life cycle of the bryophytes, you know that one. Here two stages are there. What are the stages here? The gametophytic generation. Another one is the sporophytic generation. Okay. So here two stages are there, means two generations are there in the bryophyte life cycle. That is the gametophytic stage. Another one is the sporophytic stage. So what are there here? Gametophyte to produce the sporophyte develop, and these sporophyte completely depending on the gametophyte. You know that one. Yes, gametophyte to produce the gametes means anthrozoids are there, then egg is there. Means anthrozoids is produced from antheridia. Egg is developed on the female reproductive organ, that is the archegonium. You know that one. Yes. Is clear that one?
Yes, sir.
So the gametophytes, the gametophytic phase to produce the reproductive gametes means male and female reproductive gametes, that is to fuse in the oogonium cell. Means pharyngeal canal cells are nothing but in the oogonium or archegonium. That is the archegonium after the fertilization. So after the fertilization of these anthrozoids and egg to form a zygote. After the zygote, here to develop as a sporophyte. So in the after these are two generations which are coming to one another, means germinative alternative means gametophytic phase and sporophytic phase. So this a series of sporophytic generation, discuss that one. This sporophytic biophyte in the simple a solid object radial in construction with without any appendages and lack of the self nutrition. We know that one in Rixia. So lack of the self nutrition means they are depending on the, okay, no, they're depending on the gametophyte only. There are concepts the food, seta, and capsule. Okay, but in Bryopsida, some assimilatory cells are there. Assimilatory cells, nothing but photosynthetic cells. Assimilatory cells, they are involved in the photosynthetic process. So that's why these mosses and some anthoceratopsida also. Anthoceras also, they are the partial is depending on the gametophyte. Sporophyte of these mosses are anthoceratopsida, partially depending on the gametophyte. But The Rixia, completely Rixia and Markantia means hepatic capsule members, the sporophyte completely depending on the gametophyte for food and nutrition, all those things. Okay, so this one, what happened here? It is depending on the gametophyte for food. It is wholly attached to the gametophyte. Its only function is production and dispersal of the spore. In Rixia, that one. Okay, only the Rixia sporophyte only to produce the spores. To produce the spores only. The structure of the producing organ Rixia are elaborate objective differentiated into food, seta, and capsule as higher bryophytes. Okay, the food function as anchoring means as supporting organ to observe and to fix the in the gametophytic tissue. And the seta is maybe small and longer, slender stalk-like structure, which is the helps in the spore dispersal and connecting between the foot and capsule. The capsule is varies in shape. We know that one. Some all spherical are there. Rixia in the spherical structure is there. See here. That is the Rixia completely spherical in structure. See here. This one. Okay, only in the different spherical structure, oval, cylindrical. Cylindrical is example, that is the anthoceras. Long, slender-like structure is there. Cylindrical-like structure. Yes, sir. So it is the difference in shapes are there. It varies in the elaboration, complexity. Elaboration means complexity and the maybe internal and external structure also. So according to the complexity of structure of the sporophyte, we already discussed that one. So complexity means structure of the sporophyte. Which one is the simple, which one is the complex structure? Here Rixia, see here Rixia, only the spherical structure is there. So complex, there is no complex foot and seta is not there. That only spherical capsule is present. Inside the capsule, only the spores are there. Okay. So only the only that is a simple structure is there. Yes or no? Here, then compared to Anthocera, this one is the Rixia sporophyte. Are you clear? Hello?
Yes, sir.
Yeah. Okay. Thank you. So this one is a very simple structure is there. Means based on the complexity. So based on the complexity here, we have to explain the two theories are there. Means simple to complex structure of the sporophyte. So this starts with the simple sporophyte of Rixia. This one is the simple sporophyte, which is Rixia. From Rixia to Markantia, then Pellia and Anthoceras. Finally, highly complexity of sporophyte in Funaria. Okay, so see here, the Funaria is have complexity structure of the sporophyte. See here, this one is highly complexity. See here, then compared to Rixia, so means sterile tissue is more. Okay, sterile tissue is more. Only the spores are here only, spore sacs. Centrally one columella is there. See here, this one is the columella. Only spore sacs only here and here only. Okay, the remaining all are the here. This total sterile tissue is more. Yes or no?
Yes, sir.
Yeah. Sorry. Again, how to again back? How it coming again back? I don't know. Yeah. Okay. So here only this one only means sterile tissue is present. Fertile tissue is there. Means the complexity is more than compared to Rixia. Yes or no? So based on these formation here, we have to explain the two theories are there to about the discuss and to development of the sporophyte, evolution of sporophyte in bryophyte. Okay. So what is the theory here? The first theory is the sterilization theory. Okay. Sterilization theory. So in this sterilization theory here, in the sterilization theory means the series of sporophyte of bryophyte show a natural advance in the progressing elaboration and complexity. See here.
So could you repeat that once again, sir? I will write down the definition.
Okay. Means that is the sterilization theory means the series of sporophyte of bryophyte show. They shows a natural advance in the processing elaboration and complexity. So here the
Thank you, sir.
the processing of complexity. What is the here? The simple to advanced. So the fundamental principle involved in this progressive sterilization of the potential fertile cells instead of forming of spores for the propagative purpose, they remain sterile. Okay, clear? So here means the fertile cell, potential fertile cell, it will be sterilized. Sterilization, sterilized means anybody sterilized? Hello? Okay. So here, what happened here? The sporogenous tissue, they are sterilized. Then in the fertile, fertile cells only. Means that what happened here? The sporogenous tissue only to produce the spore mother cells and to produce the some elater and remaining the sterile cells will be increased gradually. It will be progressed. So based on this one, it is explained. The sterile cells takes function of the nutrition and descent and dispersal of the spores. So means here the sterile tissues will be increased. Okay, sterile tissue will be increased and doing the other functions means sterilization functions like that nutrition, prepare the food and involve the nutritional process and spore dehiscence, spore dispersal mechanism which is involved in the sterilized tissue. Okay. In this hypothesis, Bower is known as this theory we call as a sterilization theory. So based on the complexity, structure of the complexity or formation of the complexity of sporophyte, which is gradually or a series will develop, which is observed by the Bower. So the theory we call as also sterilization theory. In evolution of sporophyte in bryophytes on the basis of the progressive elaboration and complexity of sporophyte. So, sterilization theory only in the based on the progressive elaboration of complexity. Okay, progressive elaboration and complexity of sporophyte. What is that? Based on the elaboration means the structure of the sporophyte will be elaborated and complexity is also more than compared to Rixia. Clear?
Yes, sir.
Okay. So, and other some supporter scientists also there. There is the Champels and Smith is also to support these sterilization theories. Okay. So, the sporophyte, here we can see the Rixia sporophyte. First of all, we see the Rixia sporophyte, how it is looks to. Only the zygote will develop as a sporophyte after the formation of fertilization. It increases in size, undergoes a single division, forming two cells. Okay, after fertilization, the zygote increased in size and undergoes to the a single mitotic division to form a embryo. Two-celled embryos is formed. So this embryo by repeated division to form around 20 to 30 undifferentiated cells that are formed. So after the differentiation into outer sterile cell and amphithecium and inner mass of cells, we call as a fertile sporogenous tissue. That one we call as the endothecium. Okay. So what happened here in the Rixia sporophyte development, the zygote will undergo to the one division to form a two-celled embryo. The zygote will be increased in size and increased in size and undergoes to the again number of divisions takes place to form around 20 to 30 mass of the cells. There is no differentiation, undifferentiated cells are there. So after some times it will be differentiated into two tiers are there. That is the outer one we call as the amphithecium, inner one we call as the endothecium. So that outer one is amphithecium to form a capsule layer, single layer of capsule cell and amphithecium cells is formed. Then inner one we call as a sporogenous tissue, that is nothing but endothecium. So after this amphithecium forms the single layer capsule wall. See here, only this one single layer capsule wall is there. See here, yes or no? Single layer capsule wall is there.
Yes, sir.
Yes.
Single layer. Ah, So single layer capsule wall is there. And then uh, so single layer capsule wall is there. Then endothecium to become as a archisporium. Okay, internal another inner type we call as a endothecium. That endothecium again to develop as a archisporium. So archisporium is nothing but sporogenous tissues. Okay. So after some potential spore mother cells are formed and some are the fail to produce the spore mother cells. Okay. So these are to formed as a nutritive cells. So in some pre that is the porination of elaters we call that. Okay, means before elaters forming cells are there, that one we use they are used as a nutritive cells to develop as a spore. So here there is no foot theta. Here only the single capsule is there. Okay, so the little sterilization means a little amount of the sterilized tissue is present. Mainly the potentially the fertile cells are there. Okay, clear this one, Rixia?
Yes, sir. Yes.
So here the sterile tissue is very little. Means there is no foot and capsule is not clearly observed in the Rixia. Only the fertile cells are more than compared to so the fertile tissue is more. So coming to the next one. So Markantia sporophyte. So coming to the from Rixia to see here. So Rixia to Markantia. So next one is the Markantia sporophyte. Here, the two-celled embryo divide at a right angle and four-celled embryo is formed. Okay. So two-celled means the zygote after fertilization, zygote undergoes the division to form a two-celled embryo. That embryo again divide to produce the four-celled embryo. So these divided into two types, the upper part and lower part. These lower two cells we call as hypobasal cells, which remains sterile to formed as a foot and theta. Lower tire of the cells, lower tire of the two cells to call as hypobasal cells, which is to produced as a sterile and to develop as a foot and theta. And the upper two tire cells we call as epibasal cells, means upper one. Epibasal cells, which is called as a fertile cells, and to repeat division to produce the upper part of the some theta and completely capsule will develop epibasal cell. The capsule portion is differentiated into outer amphitheecium and inner endothecium. Okay. The single layer capsule wall is formed from the amphitheecium, outer amphitheecium, to produce a single layer of the capsule wall, and the entire mass of the endothecium to act as a sporogenous tissue or archospore mother cells are sporogenous tissue. So these sporogenous tissues became the coiled thick bond and became uh, archosporium, we call that. So half of the archosporium to develop as a sterile and which is developed as a pseudoelater. Half of the archosporium cells to develop as pseudoelaters, and remaining half of the archosporium to develop as a spore mother cells. Clear? Hello? Okay.
Yes, sir.
Yes, sir.
Okay. So that is the, and we know that one, that elaters here they have the hygroscopic nature is there, and spindle-like structures, long elated spindle-like structures, spiral thickenings are there on the surface of the elater. Spiral thickenings and hygroscopic nature is present. Hygroscopic spiral thickenings are there. So these elaters which are used are involved in the pore dispersal mechanism. Okay. Ah. So these some organospores they are to convert into sterile apical cells also there. Okay. Ah. So the sporophyte of the Markantia consists of broad foot and well-developed theta and a single layer sterile jacket cells of the capsule. In this diagram, we see here the food, seta, and capsule is present. But in Rixia, there is no food, seta. Clear? There is no food and seta. And here, the sterile is more than compared to Rixia. The Markantia sporophyte sterile tissue progressively appear than compared to Rixia. Okay, Nandi, clear? Huh? So here,
Yes, sir.
the outer layer of the capsule wall is made up of a single layer is there. And the apical cap at the apex that has spiral thickening elongated, the sterile tissues are there. This one, we know that one, these are involved in the pore dispersal. And another purified, the progressive sterilization theory, another one is the Pelia. See here, see here, Pelia. This one is the Pelia. Pelia purified. In this, the zygote then undergoes to the number of division after fertilization. Here increased in size, similarly increased in size and large in size and divided into two cells, that in the lower one and upper one. The hyperbasal cells does not take place part in the development of the sporophyte. Clear? Here, the hyperbasal cells means lower tier of the cells which is does not involve in the sporophyte development. Okay, only the epibasal cells only divide and to produce into the development of the sporophyte. Means sporophyte means spore producing organ. Okay. Ah, the epibasal cells divide to differentiate into outer amphitheecium and inner endothecium. Epibasal cell only means upper tier cells only divide to produce the outer amphitheecium and inner endothecium. The amphitheecium to form the capsule wall and endothecium to act as a archisporium or spore mother cell. The upper half of the epibasal cell to form the capsule and the lower half to form as a photoseta. Yeah. These sporogenous tissue of the endothecium to form the four mother cells and some sterile elaters also developed. These elaters are long, slender with double spiral thickness on their walls. In the pelia, the elaters they have the double spiral thickness on their wall. So some sterile cells became the compact and to form the elatophores. Elatophores also form. This is used to spore dispersal. Here, the sterilization process is advanced, further forming the efficient spore dispersal. Okay. So, which is very which is very efficient mechanism will develop for the spore dispersal structure in these elatophore. Okay. This is the Pelia sporophyte. Okay, clear? Pelia sporophyte, the hypobasal cell, which is does not involve in the development of the sporophyte. Only the upper part of the tire means epibasal cell only to divide and to develop as a amphitheecium, outer amphitheecium and inner endothecium. Okay. So outer amphitheecium to develop as a capsule wall and inner endothecium to develop as a archisporium. Archisporium means only this one only to develop as sterile means a fertile tissue to develop as a porogenous porogenous tissue or spore mother cell. Okay, clear on this one, Apelia?
Yes, sir.
Yes, sir.
Okay. So here the well-developed foot and seta is there. Means see here the sterile tissue completely here sterile tissue. These are the all are the sterile tissue. Okay. Only the fertile tissue here. This one only fertile tissue means fertile tissue gradually decreased. Okay, clear. So gradually decreased means the capsule wall is also around 4 to 5 layer of the capsule wall is occur. Then compared to Rixia, Rixia only the single layer of capsule wall is there. See here, the capsule wall here around 3 to 4 layer of the capsule wall is present. Yes or no? And long seta is there, foot is also there. This foot is anchor-like structure which is embedded into the gametophyte to observe or to fix or to anchor-like structure and act as a supporting organ of this sporophyte. And seta, long, slender like seta is also there, which is made up of the multicellular around 4 to 5 layer of the multicellular stalk is there. Upon the stalk, a capsule is present, that is the oval-like capsule is there or spherical-like capsule. The capsule wall is also made up around 4 to 5 cell walls are there. Cells, 4 to 5 layer of the cells are present. Okay, clear? So coming to the anthoceras, you see here. So anthoceras. So here in Anthocera sporophyte development, the two-celled embryo is converted into eight-celled embryo. Means two-celled again to divide to produce the four-celled. That four-celled again to divide to form a eight antank or eight-celled. So from that eight-celled upper one to call as a four tires upper tire and lower four cells we call as a lower tier of the cell. So here eight-celled embryo is formed and arranged into two tiers of the four cells. In each tier, here have four cells are there. The lower tier again further divide to form to produce the massive food. Okay, massive food and the cells on upper tier is also repeated division to form a capsule. From that eight actant embryo, eight-celled embryo to form a two tiers, upper tier, then lower tier. So each tier is have the four cells are there. In the lower tier, they are to develop as a number of divisions to develop as the food, massive cell of the food. See here, a large sterile tissue of the foot is there and the upper tier to develop as the capsule. The capsule in young condition, it is differentiated into outer amphitheecium and inner endothecium. The endothecium is sterile to form the central columella of sterile cells called as a columella. We know that one. Outer amphitheecium, outer that is the amphitheecium and inner endothecium. So in endothecium is also some sterile tissues developed in centrally that can be called as a columella. The surrounding and overcharging to top of the columella, there is a sheet of the sporogenous tissue. The archisporium, which is the extremely reduced. Archisporium totally, it is some reduced is going on. So the archaeosporium forms these four mother cells, the pseudoelaters also. The archaeosporium is anthoceras is thus extremely reduced to form the narrow part of the capsule. See here, the beside the columella. See here, the only beside the columella only here. See here. Sorry, only beside the columella. This one, the centrally one columella is there. So this one central columella is there. Around 4 to 5 cells of the sterile tissue columella is there. So beside the columella, this one is a fertile sac is there. This one. Okay. Fertile sac is present. So these archaeosporium mother cells and some pseudoelaters also form. Means the fertile tissue gradually sterile means reduced. Okay. Fertile is gradually reduced means sterile cells gradually increased. So in this narrow part of the capsule, the covering layer of the capsule and archaeosporium and derived from the epithelium. The jacket layers consist of chloroplast and stomata also present. So here jacket cells means outer layer of the capsule which is made up of these some stomata also there, and parenchyma tissue means chlorophyll collenchyma, nothing but collenchyma tissue is there, the outer area of the cap. And this unable to sporophyte to lead the independent to existence. This type of the sporophyte shows marked reduction in the sporogenous tissue due to the more sterilization, means more sterile tissue is formed. Okay, clear? Anthocera sporophyte.
Yes, sir.
Means from marca means from Rixia, Marcantia, and Pelia to here Anthoceras. The sterile tissue is more than compared to the previous one. Then compared to means previous one means Rixia and Marcantia. So here the foot is there, bulbous like foot is present, which is the embedded in the gametophytic tissue, and instead of the seta, a meristematic tissue is also there. Meristematic tissue is present in this anthoceras. So the central one columella is also there. This is also the sterile tissue which is developed from the endothelium. Some part of the endothelium means and only some portion of the endothelium only to develop as a archaeosporium. Archaeosporium are nothing but four mother cells. Four producing organ or four mother cells. Archaeosporium is nothing but. Okay. The remaining centrally columella is there, and capsule wall is made up of around 4 to 5 layers of the cells are there. So that layers they have the chloroplasts are there. So that's why that one we call as the collenchyma. So which is involved in the photosynthesis, and they have the stomatas also present. Means sterilization is more than compared to remaining Rixia and Marcantia, and the fertile tissue is reduced. Fertile tissue is reduced. Okay, clear?
Yes, sir.
So next coming to the Funaria. In the Funaria sporophyte here, this one is the Funaria sporophyte. So the major part of the sporophyte remains sterile and form the foot and seta. So these total foot and seta is there. Apophysis is also there. Apophysis involved in the photosynthesis. This one is the apophysis. This one is the apophysis. Okay, this one, apophysis is there. So central one conducting tissue is there. This is the conducting tissue, and here the apophysis is involved in the photosynthesis. And major part of the sterile tissue to formed as a foot and theta. The capsule of the sporophyte is developed from the epibasal cell, and the foot and theta from developed as a hypobasal cell. Okay, hypobasal cells means lower part of the basal cell means hypobasal tire or hypobasal part to develop as a foot and theta, and epibasal cell developed as capsule. The epibasal cells again divide periclinally to form as outer amphitheecium and inner endothelium. The amphitheecium became differentiated into epidermis and photosynthetic tissue of the capsule wall and outer spore sac. Okay, that is the
Yes, sir.
amphitheecium, outer amphitheecium to develop as the these epidermis, photosynthetic tissue, and remaining spore wall. These are developed from the outer amphitheecium. The amphitheecium at the apex of the capsule to form the multi-layer operculum also developed from the amphitheecium only. So these operculum here they have the peristome teeth present in the operculum. See here, this one is the operculum opening area. This one is also derived from the amphitheecium only. So in this operculum, they have the peristome teeth. Peristome teeth is present. So these peristome teeth involved in the pore dispersal. Peristome teeth are there around 16 to 34 peristome teeth are present in the finaria. Okay. Uh, So, so peristome teeth are there. The endothelium is the sterile, the thin outermost layer of the outer layer, that is the archisporium. The capsule is pear-shaped structure with its basal one sterile, and that one we known as apophysis. This one is apophysis. See here, already explained that one. Okay. So these sporophyte, they are look like a pear-like structure is there. The outermost layer is the epidermis layer, and below the massive parenchyma cells, so they have the chloroplast, and they are doing the photosynthesis. They are involved in the photosynthesis. There are the large air space also there, air chambers also there. So this one is the air chambers. These are the air chambers are there. These all are the air chambers. So large number of the air chambers are there. This is each air chamber is divided a uh, single layer of the cell, that one we call as the trabeculae. That one is also parenchyma tissue. That one we call as the trabeculate or trabeculate. Okay. So here the surrounding the surrounding lid of the capsule, a ring of the uh, cuticularized epidermal cells are there. That one we call as the annulus is present, which is helps in the spore dispersal. Annulus, this is the annulus. A cuticized, cutinized annulus is present. Okay. So the progressive sterilization of the potentially porogenous tissue is increased in the finaria. So this one is explain the theory of the sterilization of the sporophyte in bryophyte. Sterilization of sporophyte in bryophyte. Okay, clear? This one.
Yes, sir.
Sterilization of
Yes, sir.
sterilization of sporophyte in bryophyte means sterilization theory. This theory is proposed by the Barber. Barber is a bryologist. He is explained the sterilization process means progressive sterilization process. Progressively developed sterilization tissue progressively developed and fertile tissue gradually reduced. So based on these complexity means simple to complexities formed. So based on these complexities formed, the Bower is explained the sterilization, progressive sterilization theory. Based on these evidences means Rixia to Funaria. Rixia is here simple sporophyte. Then compared to the so the Rixia sporophyte is very simple structure of the sporophyte then compared to Funaria. Okay, clear?
Yes, sir.
Now another theory is there. The theory we call as the reduction theory. Okay, in this reduction theory means reduction means here decreased. What is decreased? Here we see that one. There is another theory is there that is explained by the Kashyap and Gobel and Ivan. Reduction theory is explained by the Kashyap, Gobel and Ivan. So they are telling that evaluation is downward direction. They are to downwards direction means complexity to simple. Clear? Huh?
Yes, sir.
Complexity
Yes, sir.
complex to downwards means complexity of the funaria is explained the simple structure, simple primitive complexity of the funaria. Okay, funaria that sporophyte is telling the simple sporophyte. Reduction means the spore genotissue is reduced. Spore genotissue is reduced. So that's why he is telling to the evaluation of sporophyte in downwards by the Kashyap and Ivan Gobel. So here the series shows retrogressive evaluation. Retrogressive means the again backward. Retrogressive evaluation. There is an simple evidence here. The reduction is accompanied by the simplification of the structure of the sporophyte. On the basis of the simple sporophyte of Rixia is considered as a highly evaluated through reduced as a result of the progressive simplification. So the Kashyap is explained the Rixia sporophyte. Simplest Rixia sporophyte is considered as a highly evaluated sporophyte. Why here highly evaluated sporophyte? Only the sporogenous tissue is more. Again, remaining the progressive simplification means the Rixia sporophyte is very simple. Yes or no?
Yes, sir.
So that's why he is telling to the result of the based on the result of what is that progressive simplification means reduction of the sterilization means reduction of the sterile tissue is more than compared to the from funaria to Rixia. Simplification of the progressive simplification is occurred is based on the these progressive is explain the reduction theory. So here some steps are there to evaluation of the sporophyte. So these evidences is explain the That is the first one is the leaves of erect sporophyte were lost due to the intense isolation and desiccation. So the funaria, they have the leaves. Sporophyte, they have the leaves, but here there are lost in the rixia. And erect leafy sporophyte became attached to the gametophyte. Okay, we can see that one. And the airspace is photosynthetic and well developed sporophyte is also depending on the gametophyte. Okay. And another stomatas are first became functional in sphagnum and later on the disappearing on the Marcon shells and Jungermon shells. Okay. And another character, the simplification of the dehiscence of the apertures means dehiscence of the capsule. Very simple. The Rixia is capsule is very simple. Dehiscence are there. Only the single layer capsule wall is present. Okay. And another character is the reduction of the photosynthetic tissue in the capsule wall. Yes or no? The funaria, they have the, see here, in the funaria, they have more complexity of the photosynthetic tissue is present than compared to, see here, than compared to Rixia. Yes or no? So A is the Rixia sporophyte. Means the complexity of sporophyte is very less. Yes or no? And
Sorry. Yes,
clear?
sir.
And another, the reduction of the photosynthetic tissue in the capsule wall also decrease in the thickness of the capsule wall with the disappearing of the thickening of wall and material of the cell wall. Okay, decreased the thickness capsule wall means the rexia here only the single layer is there. Then remaining the palea, these all are the around 4 to 5 layer of the capsule wall is there. So that's why the capsule wall is also decreased gradually. It will be decreased means the sporophyte is simplification, progressive simplification is there. Reduction simplification is occur. So that's why the rexia is only the capsule wall is single layer. So then compared to remaining the palea and marcontia and other advanced
And
complexity of the funaria. Okay, funaria is also they have the sterile tissue is more. Yes or no?
yes, sir.
Decreasing the sterile tissue. And another character, these changes slowly eliminate on the seat and foot. So the foot and seta gradually disappeared. Means sterile tissue. These foot and seta, these are the sterile tissue. So that's why these are the gradually disappeared. There is no foot and seta in Rixia. So another character, all these changes happen for the progress to increase in the fertile of the sporogenous tissue. So Kashyap and Evans, these are scientists to explain what and the progressive increase of the fertile tissue. Fertile tissue gradually is increased. Here means downwards. He is explain the downwards to downwards to the development of this sporophyte. So that's why here increased the sporogenous tissue means fertile tissue gradually increased. Okay, clear? Huh?
Yes, sir.
Okay. So progressive sterilization theory, another one reduction theory. Clear is anybody? Everybody clear?
Yes.
Okay, all of you. This one compulsory coming in the essay, evaluation of sporophyte in bryophyte, you should write these two theories with the diagrams also. Each sporophyte development you should explain and with the diagrams also. And that is anybody tell based on the what characters? Sterilization theory.
Sterilization characters.
Hmm? Bambina. Raju, Bambina Raju. Who is his name? I don't know.
Elaborations and complexity of
So elaboration and
and complexity.
progressive elaboration and complexity, development of the complexity. Gradually, it is elaborate, progressive elaboration and complexity development. Based on this one, the Rixia is the simplest sporophyte and well advanced sporophyte is Funaria. Here, Rixia, there is no sterile tissue. Very limited of sterile tissue is there. Maximum sporogenous tissue. Nothing but fertile cell is there. Maximum fertile cells are there. Only little amount of the sterile tissue is present. Here, there is no foot and seta is not there. Only single layer of the capsule wall is present. These all are the sterile tissue. Yes or no? So coming from the Markantia, sterile tissue gradually increased and Pelia gradually increased sterile tissue. From that Anthocera slowly, the progressively sterile tissue is increased. Finally, the Funaria, maximum sterile tissue is occurred. This one, the complexity means progressive sterilization, elaboration of the progressive elaboration of the complexity, progressive elaboration and complexity of the sporophyte. This is sterilization theory. But reduction theory is downwards. Okay, downwards means reduced the sterile tissue, reduced the sterile tissue to increase the fertile tissue. Okay, let's talk 2 minutes and then
Yes,
continue.
sir.
Hello. Hello.
Hello. Hello. Hello.
Yes, sir.
Okay. So sorry for this man. So now clear. So this is the evolution of sporophyte in bryophytes. Okay. So here there are two well marked alternation of generation. We already discussed that one in the life cycle of the bryophytes. That is the gametophytic phase and the sporophytic phase. Okay. So here the sporophyte is developed from the diploid zygote. We know that one after the fertilization. The sporophyte of bryophyte is simple, a solid object and radial in construction without any appendages as in the Rixia. So here tricky, but well developed behind polytrype. So it wholly attached to the gametophyte. Its only function in the production and core dispersal. So here there are the two main theories are there to explain the evolution of sporophyte in bryophyte. So first one theory we call as a sterilization theory, which is proposed by the Bowe. The progressive sterilization of the potential fertile cells. Progressive sterilization of the potentially fertile cells. So this is the fundamental principle in the sterilization theory. So some other supporters also there in the what are there here to explain the sterilization theory. That is the Cavers and Campbell Smith. This is also other scientists involved in the explain the other supporters to the progressive sterilization. What is the progressive sterilization theory principle here? Progressive sterilization of potentially fertile cells means sterile cells gradually developed instead of the fertile cells, potentially instead of the fertile cells. Fertile cells involved in the tissue means core producing organ. In sterile tissue, they are not involved in the core cells. Involved in the nutrition and other photosynthetic process. So another theory, second theory, is the reduction. Also the Kashyap and Eva. So in this theory, is explain the downwards direction. That is, the reduction is accompanied by the simplification of the structure of sporophyte. Okay, simplification of structure of sporophyte. So is explained this based on this character. So which one is the simple? Structurally, which one is the simple? Is explained. Simple sporophyte. Okay, clear? Okay, Nandi. You respond. Anybody respond?
Yes, sir.
Okay, clear? Huh?
Yes, sir.
So these two theories to explain the sporophyte in evolution. So this one, sporophyte evolution in bryophyte, is compulsory asking question in essay question. This one compulsory asking, and every time this one is coming in the examination point of view. So
Okay,
that's why you should write the two theories. That is the sterilization theory. Another one is the reduction. Reduction theory. You should write, and diagrams also. Diagrams also. And funeria diagrams also you should draw. Okay. So.
Okay, sir.
So any doubt?
No, sir.
Hmm?
No doubt, sir.
No. Okay. Thank you for attending the class.
Thank you, sir.
Few minutes you wait.
Okay, sir.
Wait. Then after go again.
Okay, sir.
Few minutes. So then 7:30 is there, so that's why only the 5 minutes you are stay in the class. After that, we have to disperse. Thank you. Okay, Andy. Thank you everybody to attend the class. Thank you so much. Tomorrow, hello. Okay.
Thank you, sir.
Thank you, sir. And again, tomorrow we explain another topic. Okay.
Okay, sir.
Thank you.
Thank
Thank
you,
you,
sir.
everybody. Now clear this evolution of sporophyte. Is any doubt?
Sir, evolution of our sporophyte is sir, evolution of sporophyte.
Sorry, not clear.
Sir, sporophytic evolution is sir important for examination, sir.
Yes, definitely. This one is most important in examination point of view. Okay.
Okay, sir. Thank you, sir.
This compulsory question.
Yes, sir. Thank you, sir.
Right. Thank you. Right. Thank you, everybody.
Okay.